Hey guys, this is Ajmera, and this video is one of multiple in a series covering topics in AP Biology. In this video, we will be covering biochemistry or biochem. Why biochem? Why is biochem so fundamental to understanding the rest of AP Biology? Well, that is because it shows itself in bits and pieces throughout the entire course, such as neurotransmission, protein synthesis, DNA mutation and replication, and also evolution. To start off, let's talk about carbohydrates, more affectionately known as carbs. Carbohydrates are the body's primary source of energy, and they come mainly in three different forms: monosaccharides, disaccharides, and polysaccharides. Monosaccharides, meaning one sugar, are the most readily available to use for energy due to the fact that they are singular. Due to their polarity, they can easily dissolve in water and blood, as can disaccharides and polysaccharides. When looking at these sugars in their structural formulas, you mainly see hexagons and pentagons, which indicate the number of carbons that sugar has in its ring. Monosaccharides with 6 carbon rings, looking like the one on the left, include glucose and galactose. Monosaccharides with 5 carbon rings, like the one on the right, include fructose. When two of these molecules combine via dehydration synthesis, a concept that we will talk about later in the video, it forms a disaccharide. Some examples of disaccharides include maltose, which is composed of 2 glucoses, and sucrose, which is formed by a glucose and a fructose. Disaccharides are often used in plants because of their ability to easily dissolve in water due to a high amount of OH- groups, which makes them very polar. The last and most complex kind of sugar is polysaccharides, which are long chains of sugars. There are 4 common types of polysaccharides. Amylose, which is found in plants, is used as a short-term storage molecule for energy. Glycogen, serving the same purpose, is found in animals. These two kinds of polysaccharides are digestible because they are made of alpha-glucose isomers that form 1 to 4 linkages. Cellulose, which is found in plants, is a structural carbohydrate and it composes most of plant cell walls. Chitin is another structural carbohydrate and it makes up exoskeletons of arthropods and fungi. These are both indigestible because they are made of beta-glucose isomers whose bond angles cannot be broken down by enzymes. Now let's move on to lipids. Lipids are the body's secondary source of energy and they are hydrocarbons, which causes them to be nonpolar or hydrophobic. The first type of lipid we will be talking about are triglycerides. Triglycerides, contrary to the name, are formed of 1 glycerol and 3 fatty acids, or fatty acid chains. The best way to imagine them is as the letter E, with 3 prongs of the letter E being the fatty acid chains and the backbone of the letter being the glycerol molecule. Its primary use is for long-term storage due to the fact that it has many carbon-hydrogen bonds. There are 2 types of triglycerides. Saturated triglycerides have every carbon in the fatty acid chain bonded to its maximum amount of hydrogens, thus no carbon double bonds. They are very straight and rigid, thus making them solid at room temperature. Unsaturated triglycerides, on the other hand, have 1 or more double bonds between carbon atoms, thus creating kinks in them, which causes those triglycerides to be liquid at room temperature. If we look at this molecule on the screen, it is clearly saturated because if you look at all of these individual fatty acid chains, they're completely saturated and you don't see any double bonds between. Phospholipids, the next type of lipid, contain a polar phosphate head, a glycerol body, and 2 fatty acid tails. If you notice, one is unsaturated and one is saturated. Phosphate head is polar, while the 2 fatty acids and the glycerol together are nonpolar. Having these 2 properties, the molecule is then considered to be amphipathic. In a water-based solution, the fatty acids of the phospholipids are attracted to each other, and the polar ends of these phospholipids are attracted to the water and each other. This allows a bilayer to be created, which also allows for a cell membrane to be created as well. The next type of lipid is terpenes, which are generally attached to a polar head, which is not depicted in here, while they themselves are nonpolar carbon chains. These molecules act as an anchor to polar molecules such as chlorophyll or other porphyrins. The fourth type of lipid is prostaglandins. Prostaglandins have a carbon ring, which is right here, followed by 2 carbon chains. These molecules are used to stimulate smooth muscle and also have an effect on uterine contraction and vasodilation. These molecules are good for being messengers because they are dissolvable in a cell's dominantly nonpolar membrane. The last type of lipid we will be talking about is steroids. Some examples of common steroids include estrogen and testosterone. Steroids vary in shape constantly, so here's a picture of cholesterol. Steroids, like prostaglandins, can readily dissolve in cell membranes and thus have an easy access to a cell. One purpose of steroids is to increase the flexibility of the cell membrane, as unsaturated steroids can help the membrane from getting too rigid in the cold. Now let's move on to proteins and amino acids. Proteins are complex macromolecules that are composed of amino acids. There exist 20 naturally occurring amino acids, and there are 2 to 3 artificially created ones. Amino acids are bonded together by peptide bonds. These peptide bonds are formed by the same processes that the covalent bonds are formed within carbohydrates, lipids, and nucleic acids. Let's look at an amino acid close up. In every amino acid, there is a central carbon. This carbon is attached to all the other 4 groups that exist in an amino acid. The group on the right is the carboxylic acid group, and the group on the left is the amino group. The amino and carboxyl groups of different amino acids are what form the peptide bonds. Under this carbon is a hydrogen atom, which is always there in all amino acids. And here is the R group, or the variable group. The variable group is the group that remains inconsistent between the 20 amino acids. Now let's talk about protein folding. Proteins can fold in many different ways depending on the DNA sequence that created them. The first level of protein folding is called its primary structure. The primary structure of a protein, as shown here, is just the sequence of the amino acids, nothing more than that. The next is secondary structure. There are 2 main forms of secondary structure. On the left, you'll see alpha helices, and on the right, you'll see beta pleated sheets. These dashed lines in the diagrams represent hydrogen bonds, because that's what secondary structures are primarily composed of. Alpha helices have a coil-like structure that goes about them. Beta pleated sheets, on the other hand, are made of beta strands that are all parallel to one another. When looking at a protein, beta pleated sheets will actually look like an actual sheet that's consistent throughout the whole protein. The tendency of one secondary structure to form over another is just dependent on the amino acids that are present in the primary structure. The next level is tertiary structure. Tertiary structure is primarily determined based off of if the amino acid is hydrophilic or hydrophobic. In a water-based solution, hydrophobic amino acids will tend to curl towards the inside of the molecule, while hydrophilic amino acids will tend to go to the outside of the molecule. However, if the protein is completely submerged inside of a membrane, which is predominantly nonpolar, the hydrophobic or nonpolar amino acids will tend to go towards the outside of the molecule, while the hydrophilic or the polar amino acids will tend to go inside the molecule. The next and final level of protein structure is quaternary structure. The quaternary structure of a protein is determined by subunits of protein chains combining together to form one piece. These pieces are held together by hydrogen bonds, charges, and also many other different bonds that I'll talk about in the protein video. There are many functions of proteins in your body. First is to act as an enzyme. Enzymes are proteins that catalyze reactions, or in other terms, decrease the amount of activation energy required to perform a reaction. It's critical that the structure of these enzymes are correct because the enzyme has to fit the substrate, which allows for the reaction to even occur. Antibodies are proteins that need to be able to bind to antigens in order to identify them to your body's immune system. It's critical that antibodies are the correct shape because if they are not, they will not be able to attach to the antigen, thus not telling your body if there's a pathogen present or not. Proteins can also act as gates or channels for ions to move across the cell membrane. This is very critical because your cell membrane is predominantly nonpolar, which does not allow charged ions or even polar molecules to go through. Protein gates and channels can allow this to happen. Motion. Proteins are critical to motion in some bacterium because Cilia and flagella are primarily composed of proteins. Hormones and hormone receptors, along with neurotransmitters, are often made of amino acids or proteins. It's critical that the shape is correct, otherwise signals cannot be sent throughout your body. The last common function of proteins in your body is structure. Hair, for example, is made of protein. A large portion of your skin is also made of protein. It's critical that these structures are correct, otherwise it can cause abnormalities in skin or hair or other structural features of your body. The extracellular matrix throughout cells is also another example of how protein can affect structure. The extracellular matrix between cells is generally composed of proteins and carbs. If the protein structure is not correct, cells will not be able to adhere to one another. As I said before, I'll be making a separate video that covers proteins only because there's much more to proteins than AP Bio does cover. Now let's move on to nucleic acids. Nucleic acids are the monomers that make up complex information storing molecules such as DNA deoxyribonucleic acid and RNA ribonucleic acid. There are three components to all of these nucleic acids. The first is a nitrogen base. In DNA, there are thymine, adenine, guanine, and cytosine, while in RNA there are adenine, guanine, cytosine, and uracil. Both of them have the same phosphate group, and DNA has a deoxyribose sugar, while ribose has a ribose sugar. The sugar in DNA is called deoxyribose because it is missing an oxygen that a ribose molecule would have. Out of these 5 nitrogen bases that you see here in this table, 2 of them are purines and the other 3 are pyrimidines. The 2 with 2 carbon rings, adenine and guanine, are considered purines, while the ones with only 1 carbon ring are considered to be pyrimidines. Now, how do we know which nitrogen bases bond with which nitrogen bases? It's quite easy to remember. Adenine, A for simplicity, bonds with either thymine or uracil. It bonds with thymine in the case of DNA, and it bonds with uracil in the case of RNA. Guanine, on the other hand, bonds with cytosine, and there's no alternate for cytosine in RNA. Adenine and thymine, or adenine and uracil, form 2 hydrogen bonds, while guanine and cytosine form 3 hydrogen bonds. I will be talking more about DNA and how these nitrogen bases, sugars and phosphates combine to make the overall DNA molecule, but that'll be in a separate video. Now let's move on to dehydration synthesis and hydrolysis. By this point, you may have wondered, or may have not, how these macromolecules are exactly formed. The process that these molecules go through when forming polymers of themselves is called dehydration synthesis. This is an example of dehydration synthesis occurring between 2 amino acids to form a dipeptide bond. What is not shown here on this diagram, however, is that there is an enzyme required to perform this reaction, otherwise there would be a large amount of energy needed. The hydrogen from the amino group of one amino acid and the OH or the hydroxide from the carboxylic group of the other amino acid combine to make water. When this occurs, a peptide bond, which is covalent, is made between the carbon and the nitrogen of the 2 amino acids. Now let's talk about the opposite of this reaction, hydrolysis. Hydrolysis is how molecules break down into their monomers after being polymers. Like dehydration synthesis, this reaction also requires an enzyme to catalyze it. Let's look at a dipeptide again, just as a better reference. If you see this peptide bond is highlighted in red, with the addition of H2O or water to this hydrolysis reaction, the negative oxygen ion is added to the carboxylic group of one amino acid, and the 2 hydrogens positively charged are attached to the amino group of the other acid. The reason why one is positively charged and the other one's negatively charged is due to the fact that H2O is polar, which means that the oxygen would have been pulling on more electrons than the hydrogens would have been. This is also why the amino acids do not look like how I may have represented previously in the video. Also keep in mind that a hydrogen is just a proton and an electron, and a difference of an electron can make it look different on a diagram. These two reactions go hand in hand because your body frequently builds up and breaks down macromolecules as it needs them. Now let's move on to biochemical evolution. Biochemical evolution is critical to understanding biochemistry, as is history to understanding contemporary social sciences. Scientists for the past couple hundred years have been speculating exactly how macromolecules and even micromolecules could have formed under primitive Earth conditions. By this point, all scientists have agreed that a certain set of conditions must have existed at primitive Earth to allow these molecules to form. The first is that there must have been a lot of heat. This heat could have been coming from volcanoes, radioactive decay, flowing magma, ultraviolet radiation, etc., etc. Most scientists also agree that there must have been an abundance of carbon in the atmosphere, coming from molecules such as CO2, CH4, etc. In addition, there must have been a source of hydrogen. This is because hydrogen can readily donate electrons, which allows for creation of more complex compounds. Two scientists named Miller and Urey created the Miller-Urey experiment to help see if these interactions could have actually occurred in a primitive Earth. In the design of this experiment, Miller and Urey put a mixture of gases that they would have thought to be in the primitive atmosphere, such as CH2, CH4, nitrogen gas, NH2, and they exposed them to lightning simulation with electrodes. After these molecules were exposed to discharge caused by electrodes, they would then go into the condenser and mix with the water. This water would be key to a lot of the reactions because, like as we've seen in cellular respiration and photosynthesis, water plays a key role in the creation and breakdown of organic molecules. This condensed liquid containing complex molecules would then get into a kind of tub where the water would be heated. This heated water would allow convection and would allow the molecules to move more and more, which promotes mixing. And once the water gets heated enough, it goes and turns into water vapor, which then goes back into the gas chamber where there is a lightning simulation. Over here, as circled, there is a stopcock for a testing sample. As the weeks went on in this experiment, these two scientists started to find amino acids, basic ones. They started to find lipids. And again, remember, these were all basics. These were not as complex as the ones that we would find today. And they found carbs. They didn't find that many complete nucleic acids because of the fact that nucleic acids are much more complex than amino acids, lipids, and carbs. This experiment helped confirm a lot of the theories and speculations that scientists had about early Earth and biochemical evolution. Now, as a review, let's look at some organic molecules and classify them by looking at their groups, looking at their structure, looking at what bonds they have. Okay, let's do a quick review of what was taught in this video. Looking at the first molecule, what kind of biochemical molecule is it? Okay, if you guessed amino acid, or if you guessed lipid, or if you guessed nucleic acid, you are wrong. It is, in fact, a carbohydrate. Now, how would you know if something is a carbohydrate? You count. Let's look at the number of carbons in this molecule. There's 6 carbons, so C6. How many hydrogens we got? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 hydrogens. And how many oxygens? 1, 2, 3, 4, 5, 6. C6H12O6. That's the formula for glucose or fructose or galactose. Just as a rule of thumb, all carbohydrates have the ratio of CXH2XOX. Not all carbohydrates that you would see always appear in the ring-like form that you tend to see in most diagrams in textbooks. Now let's look at this next molecule. This molecule may be a little bit harder to identify, but let's see if we can figure it out. Okay, if you guessed protein, then you are right. But how do we know this is a protein but not DNA? Because it seems to be going up in a coil-like fashion. We know this is protein because we see these R groups. The R group, just like in the main structure of a protein, represents the variable group, which is what changes between the 20 amino acids. Specifically, what kind of structure is this in a protein? We know this is a secondary structure we're looking at because there are hydrogen bonds indicated. Now, again, how do we tell that it's an alpha helix? Remember how I said before that this could be confused by DNA. Since DNA also makes a helix, it's safe to assume that the structure that we're looking at here is an alpha helix protein. Let's take a look at the next molecule. Okay, just as a hint for this molecule, I would try to think of it as 3 separate parts of the molecule rather than the entire one, because that could be a little bit confusing. So let's see if we can figure it out. Okay, if you guessed nucleic acid, you are correct. Now, how do we know that this is a nucleic acid? Well, as I kind of gave in the hint, there are 3 different parts to this nucleic acid. We have our phosphate group here, we have our ribose sugar or deoxyribose sugar here, and this question is irrelevant, and then we have our nitrogen base right here. We can tell that it's a phosphate group because it's the only part of the atom that is phosphate. The nitrogen group is identifiable because, well, of all of the nitrogens, and the ribose group is identifiable because of the fact that it makes a carbon ring, much like other sugars. To be specific, this nucleotide is thymine, but you will not necessarily need to know that for AP biology. Now let's look at the next molecule. Okay, if you guessed fatty acid, you are correct. But how do we know that it's not a carbohydrate, considering that there are oxygens right here? Well, if you look at the ratio that I mentioned before, C to X, H to 2X, and O to X, this molecule does not follow that pattern as there are way more carbons and hydrogens than oxygens. So this does not apply here. But how do you know this is a fatty acid in particular? Well, if you look at the traditional triglyceride, looks like a E, where this is the glycerol, and these are the 3 fatty acid chains. You would notice that this bonds with the glycerol at these intersections. Now let's move on to the next molecule. Okay, let's see if we can figure it out. Okay, if you guessed steroid, you are correct. So we know it's a lipid because it has these carbon rings that do not have the same bonds that carbohydrates have between each other. These carbon rings are characteristic to steroids, such as testosterone, cholesterol, estrogen, progesterone. This one specifically is testosterone, but again, you will not need to know that necessarily for the exam. How else can we tell that this molecule is a steroid? Well, prostaglandins have carbon tails kind of going out from them. Phospholipids look pretty much the exact same, except they have a phosphate group, a glycerol, and then fatty acids. And we know it's not a triglyceride because triglycerides have an E-looking shape to them. And we can tell that it's not a terpene because a terpene is just along hydrocarbon chain. Okay, thanks for watching, and be sure to check out the preliminary chemistry video if you do not understand some of these concepts of bonding. And if you want to move on to the next topic, we will also be posting a video on cellular biology. Hey guys, thanks for watching. If there's any part of the video that you missed, there are links on this screen. Make sure to like and comment on this video and be sure to also subscribe to this channel.